Right, so hello everyone. Thought we'd start promptly at 9:30. Hopefully everyone can hear me okay and I'm not sounding too loud. So I'm here, my name is Dr. Christian Wood, and I'm going to be co-chairing with Miss Victoria Esewoku on this symposium today hosted by the British Association of Psychopharmacology, looking at the role of corticostriatal circuits in schizophrenia and depression. And obviously huge thanks to the BAP for allowing us to have this session here at BNA this year in Liverpool. So just to give a little bit of a background as to who the BAP are and why they're kind of funding this symposia. So the BAP is one of the largest national societies studying psychopharmacology and neuroscience related areas. The largest in terms of national society in Europe and kind of the second largest globally. And really the BAP is a very open and supportive and importantly democratic organization that really does support people at lots of different levels, from the undergraduate through to PI, senior PI and beyond. And then they're very, very generous in terms of travel awards for their annual meeting, which kind of dots around the UK. And then critically, they have a lot of different prizes and awards at lots of different career levels from, say, junior undergraduate all the way through to, say, junior PIs, but then also up to senior PIs too. And then in terms of the BAP, it's not just about research. They do a lot in terms of education for individuals and continuing professional development from both a non-clinical and clinical perspective with things like masterclasses, integrating information across both academia and also industry. And as I said, they have an annual meeting, and I was slightly worried about putting this picture up here in the top left about this year's meeting because it's from the other place, knowing we're here in Liverpool. But importantly, a lot of stuff related to this year is kind of finalized in terms of abstract submission and things like that. But one should consider perhaps joining us next year, where we're in the middle of the country in Birmingham. It's generally late in July the summer meetings are. So perhaps when there are no BNA meetings, it might be worth thinking about joining us, the BAP. An important thing to point out in terms of these summer meetings that the BAP hosts is that they are a really balanced representation of not just clinical work, but also non-clinical work, integrating things like neuroscience aspects related to psychopharmacology. But importantly, for those of you who may be more junior in the audience, they have a lot of initiatives to kind of help to build your network and create a really inclusive environment, which is quite fantastic, with things like a buddy system and things like this, which really help you kind of build those support networks. And with that then, we'll start off with our first speaker here in this session, Dr. Madeline King, focusing on the effects of psychosocial stress on cortical GABAergic interventions and inflammatory markers in single versus dual hit neurodevelopmental models. Thank you, Maddie.
Thanks, Christian. So yes, a lot of the work I'm going to be talking about today is based around psychosocial stress and social isolation rearing of rats. But recently we have started to combine that with a second hit, which is neonatal PCP administration. So just a brief run through of our isolation rearing model. This is something that's been ongoing in Nottingham for many years, since sort of the early 1990s with my former PhD supervisors, Charles Marsden and then Kevin Phone. And how we go about it is when the rats reach 3 weeks of age, which is the point that they naturally could become independent from their dam, typically people will house the animals in social groups of 3 to 4 rats per cage. But for our isolation rearing model, we place them individually, so in cages on their own. And they are maintained under those conditions for the rest of their lifespan, so we don't re-socialize them at any point. And we're quite careful of how we interact with them as scientists and what we do with them. So we don't handle them very much at all. They just get a single weekly bedding change. And then the only other times we interact with them would be during any dosing or any behavioural testing. So they have a minimal handling. But importantly, they do have bedding in the cage, sawdust bedding, ad-lib food and water. And they can see and smell and hear other rats in adjacent cages. So if you think to more recent times, when we were allowed to interact online and so in a socially distanced way with those outside our immediate family group, this is perhaps actually quite translationally relevant. And in terms of the rat lifespan, we're allowed to isolate these animals for up to 16 weeks in terms of our ethical and legal permissions. And if you translate that to human terms, that's about 3.5 years. So it is plausible that those who've been through, a global pandemic and perhaps have other family circumstances going on, bereavements, and things like that, might experience something on a similar scale. And crucially, the animals will develop a full array of behavioural, structural and neurochemical deficits which they emerge in early adulthood, and they're not restricted to any one particular neurotransmitter pathway. So it quite maps complex disorders like schizophrenia and depression, which are not disorders affecting a single receptor or a single neurotransmitter or even a single brain region. So, in a nod to the symposium title, I thought I'd better talk about the cortex and some of the work we've done in the striatum. So, previous work in Nottingham using structural MRI imaging has shown that the isolation-reared rats have about a 5% decrease in the volume of their prefrontal cortex. And some work that we did in collaboration with Phil Seaman showed if you take the striatum and homogenise this, the isolation-reared rats have a higher proportion of their dopamine D2 receptors, which are in a sort of a higher affinity state for dopamine. So there's something going on with striatal dopamine, and they also have a smaller prefrontal cortex. And in vivo microdialysis has shown that within the striatum, the isolation-reared rats have an increased response to amphetamine. So, on the graph here, these are the control animals challenged with amphetamine, and social isolation-reared rats have a much bigger dopaminergic response to amphetamine challenge. And that matches what we know from human PET imaging, where individuals who have schizophrenia and receive an amphetamine challenge show a greater dopamine response than control individuals. And turning back then to the frontal cortex, we've done some transcriptomic work, and I'm going to elaborate in this a little bit more in my presentation at the BAP meeting this summer. But we basically looked at transcriptomic changes in the frontal cortex of isolation-reared rats, and then we matched those with published human data sets from different disease states. And you can see an overlap both with schizophrenia and major depressive disorder, bipolar disorder, and in the middle, transcriptomic changes in the isolates that overlap with multiple disease states. So this all sounds quite positive, and you think, okay, there's things going on in these animals that would match depression, schizophrenia-like states in patients. But a note of caution. So we've used these animals as a tool for some of the early preclinical evaluation of various test compounds, and sadly, the model doesn't necessarily perform quite as we would want, in that a lot of agents will reverse various behavioural deficits, so novel object discrimination, social interaction, pre-pulse inhibition of startle in our animal model, but when these compounds pass to clinical trials, they're not effective, or at least not to the extent that one would want. So what do we do about this? Well, we decided to try and adopt a dual hit approach, combining the isolation rearing with other hits to try and make a model with better predictive validity. We initially looked at maternal immune activation, got some very unexpected findings there in that those two particular hits made the animals more resilient. So we didn't actually see any behavioural changes that you could progress to drug reversal studies. So we then switched to neonatal PCP administration, which is a widely established model in its own right. People are using neonatal administration of NMDA receptor antagonists as a single hit, and that does produce a behavioural spectrum, structural neurochemical deficits. So we adopted this into our dual hit model. So we give the animals PCP on postnatal days 7, 9, and 11, and this causes a lasting excitatory inhibitory imbalance. When they get to weaning age, they're either vehicle animals would be group housed, PCP-treated animals would be socially isolated, so we have vehicle group house controls and PCP isolate dual hit animals. Clicker has stopped working. Okay. And just as with the isolates, the deficits emerge in early adulthood, but they are more pronounced in the dual hit model than in the single hit model. So we see a wider spectrum of social deficits and more changes in reversal learning as well. And the evidence that we've got so far suggests this dual hit model has much better face and, importantly, predictive validity. So this is a summary of the test compounds we've evaluated so far in the dual hit model. 5-HT6 receptor agonists do reverse the behavioural changes in the single-hit isolates. They aren't effective clinically, and I'm pleased to say they also are not active in the PCP isolate model. We've looked at the voltage-gated sodium channel blocker, lamotrigine, which is used in the management of some clozapine-resistant schizophrenia cases. We've looked at the D2, D3 partial agonist cariprazine, GlyT1 inhibitors, and muscarinic compounds. And in as much as is possible, we think that the changes we see in our animals, the reversals that we see, match the clinical trials of those compounds. We have looked at mGlu7 compounds, but there's no clinical data on that yet, but we think that would be an interesting area. So that's a bit of background. So what I'm going to talk to you today is about some immunohistochemical examinations in the prefrontal cortex of the PCP isolate animals compared to the vehicle isolate animals and the group house controls. And we initially focused on parvalbumin positive interneurons in the frontal cortex and also perineuronal nets. And I know a lot of the audience will be familiar with that. I've seen a lot of really nice posters on that work yesterday, those markers in other animal models. We also looked at microglial activation because there are changes in that in individuals with schizophrenia. So we focused on a marker called IBA1 and also looked at oxidative stress where we used a marker called 4-HNE, 4-hydroxynonenal, which is a marker for lipid peroxidation and crucially has been shown to change in patient populations. We've also looked at oligodendrocyte myelination markers, so something called CNPAs, and in a nod to Oliver Howe's work on synaptic vesicle markers in patient populations, we've also looked immunohistochemically at SV2A, synaptic vesicle 2A protein. So I'm just going to run you through the data for each of those markers now. Before I do that, I just want to explain the orientation of my graphs. So we looked at the ventromedial and the dorsolateral orbitofrontal cortex, and also a pooled region that spans the prelimbic and the infralimbic cortices. And the left hand, the open bars, is always the vehicle group house control, the middle vehicle isolate, and the right hand in each set, the dark grey, is the PCP isolates. So same layout on all the graphs. With regards to parvalbumin, so our marker for a particular subset of GABAergic interneurons, we see a reduced intensity and count in the prelimbic cortex of both the vehicle isolates and the PCP isolates. But we also see decreases in the orbitofrontal cortex in the PCP isolates only. So we're seeing something extra going on in those animals, and that pattern is really repeated throughout most of the data I'll show today. We didn't see changes to calbindin or somatostatin positive interneurons in either of the models, but we did see changes to numbers of perineuronal nets. So these are a specialized form of extracellular matrix which typically surrounds parvalbumin positive interneurons. In the ventromedial prefrontal cortex or orbitofrontal cortex, reductions in the numbers of these structures in both models. But when we actually looked at the nets in more detail, sort of the morphology, if you like, the quality of them, we looked at the thickness of the staining surrounding each cell body. And we see that although in the prelimbic cortex there are decreases in both models, in the orbitofrontal cortex we're only seeing a reduced thickness of this staining in the PCP isolate dual hit model. When we looked at inflammatory markers, so microglial activation, this is staining for IBA1, and you can see the cells typically have a sort of a resting morphology, but in the PCP isolates, they're transitioning towards a more activated state where the nucleus becomes elongated, rod-shaped. This is increased activation state and occurs only in the PCP isolate animals. We also, this is the one piece of data that's not immunohistochemical, we measured the inflammatory cytokine IL-6 just using ELISA in bulk tissue, so that's why I don't have the subregion definitions for you on this one, but we did see an increase in cytokine levels only in the PCP isolate animals. And then looking at our lipid peroxidation marker for hydroxynonenal, that was increased also only in the PCP isolate animals. And maybe this hasn't come out too well, but you see a sort of a halo of staining around the cell membrane, and that typically is increased in the PCP isolates. And then moving on to our oligodendrocyte markers, so CNPase is a marker for myelination. And again, these images haven't come out. They looked fine on my computer at home, but we see a reduced staining for that marker in the orbitofrontal cortex of the PCP isolates, but no change in our single-hit vehicle isolates. So we were expecting, based on all these changes, that we would also see a decrease in expression of synaptic vesicle protein 2A. And when I looked at the data in more detail, I think there's a hint that there may be some subgroups of PCP isolates that are responders or non-responders, particularly within the anterior cingulate cortex. I've deliberately not shown that data today because I think it's quite preliminary. I would prefer to replicate it in another cohort of animals before I'm confident in it. But in the regions I've discussed today, overall, no significant change in the SV2A staining. So, to summarise all that data, in the vehicle isolates, we see some subtle changes in the prefrontal cortex, in the parvalbumin interneuron counts, and in some of our perineuronal net measures. But when we incorporate the neonatal PCP before that, we see a much wider spectrum of changes, particularly in the orbitofrontal cortex. So we're seeing reduced thickness of perineuronal nets, increased microglial activation, increased cytokine levels, increased lipid peroxidation, and reduced staining for myelin markers in oligodendrocytes. And the orbitofrontal cortex is a region that connects intimately with the striatum, so you might think that hopefully you could go on to study frontocortical striatal connectivity in this model. We haven't done that yet. I'd be very open to opportunities for collaborations in that space. But I think that's a great way that we could move this forward. So in conclusion, I think the work that I've shown, trying to put it together into a framework or a flow diagram of the order of events, I think that the things we've shown could very easily align with the work of Kim Do from Lausanne. So giving neonatal, or PCP in the neonatal period could promote a redox dysregulation, which in turn has a negative impact on oligodendrocytes, on the perineuronal nets, and promotes microglial activation. In turn, these three events have negative consequences for the parvalbumin-positive interneurons, so these fast-spiking, very active neurons within the prefrontal cortex. And that, in turn, that disruption of the parvalbumin interneurons will feed back and cause an excitatory inhibitory imbalance, which will add to that caused already by the neonatal PCP administration. Our isolation rearing, we know, can promote perineuronal damage in and of itself, and also the change in parvalbumin expression. So it can add to the consequences of neonatal PCP administration. We haven't seen so far robust evidence of the aberrant synaptic pruning. It's not to say it doesn't happen. It may just be that these particular cohort of animals, perhaps the tissue was collected a little bit too early, and if we'd left them a little bit longer, we might have seen that emerge. So I said we can do this for up to 16 weeks within our legal permissions and our ethical permissions. This particular cohort of animals was terminated around 8 weeks. So maybe if we'd run them on for a little bit longer, we might have seen the full spectrum of immunohistochemical changes emerge. So I think this is an exciting model for us. We would like to continue using it for future evaluation of novel test compounds. We think it encompasses a wide range of pathology that we could look and see whether the drugs don't just affect behavior, but also reverse some of these proteomic changes. And I'd really like to see this model included in the list. There was a great session yesterday about animal models and creating a catalogue of models. I'd love it if we could get this included, and it can spread and be used sort of a bit more widely beyond Nottingham. So I'd just like to thank those who contributed to the work I've presented today. So Jen Cale was a former MRes student in my group, continued on to a PhD elsewhere in Nottingham, and she sectioned the frontal cortical tissue and did the GABA interneuron work and the microglial activation work with a team of final year project students. Victoria, our co-chair, did the SV2A work and the 4HNE work during a one-year internship in the second year of her degree. Jingjing Feng, an undergraduate student who was funded by a BAP internship, and she did the CMPA's work over the summer between her first and second years. And then a team of undergraduate students led by Sarah Ebegbolam did the perineuronal network, and that's Sarah actually presenting her work at the BAP last year, where she got one of the undergraduate poster prizes. So if you do have undergraduates in your group, I would say please send them to BAP. It's a great place for undergraduates to present their findings, and I will leave it there. I'd be happy to take any questions. So if we could just wait for the microphone.
I can try and speak up.
Yeah, if you speak and I'll repeat the question.
Okay, that's great. So you suggested that you added the PCP challenge to enhance the predictive validity of your model. And then you showed 3 groups, quite often, but not the 4th group, which would be
Absolutely.
PCP alone. So I was wondering, for which of your phenotypes is the addition of the isolation stressor the necessary requirement? Or could you just get all the phenotypes from PCP alone?
No. So the reason, good spot, that we don't always have the PCP group-housed control in there. We did have that in our initial studies, which have been published. I didn't include them here because they were not the most recent data. PhD student Phil Gaskin in about 2014, and some work I did with some glutamate microsensors about 10 years ago as well. And we see that actually, although people do use neonatal PCP and other NMDA antagonists as a model in their own right, we didn't see very much phenotype from those animals at all. So when we looked at novel object discrimination, which is robustly impaired by isolation, those animals didn't have that cognitive deficit. And ethically, we didn't feel comfortable to keep including a group of animals that we're neonatally treating, and financially as well, and in terms of all the other consumables, if at the end of it, they don't show much of a phenotype. So we did have that group in some of the earlier studies, but we've stopped to try and comply with the 3 Rs. And yeah, so I think a lot of the phenotype is driven by the isolation, but then with the things I've shown today, adding the PCP enhances the isolation effect. I can't say for, for parvalbumin, for perineuronal nets, etc., what PCP alone would do. A question there, the lady in pink, if she could have a microphone.
Hi. Oh, sorry, can you hear me?
Oh, yeah.
I'm just wondering how long-lasting the isolation effect is. So if you group house them again at the end of the 16 weeks, do they recover any of it, or do they keep the...
Yes, good question. So we have not re-socialized these animals. There is a lot of literature on people doing isolation and then re-socialization, but a lot of that literature is not what I would call isolation rearing. It's social isolation, so the animals reach adulthood and then they're socially isolated, typically for a period of maybe 3 weeks, and then they'd be re-socialized for a couple of weeks and then assessed. And a lot of those changes are reversible. With our isolates, we haven't re-socialized them, and to be honest, I would be a little bit worried. I'm happy that we do social interaction testing with them when we put 2 animals in an unfamiliar arena and we watch them and we video them for 10 minutes. If anything untoward, aggressive were to happen, we could separate them. I would be less comfortable, I think, to put them in a cage together and go home at night, because things that animals do in one phase of the circadian cycle, behaviour can be markedly different in the opposite phase. And I mean, maybe a sort of partial resocialization, so a cage with a barrier, so that they can maybe touch, but there's a safe escape if they want. Could be something to look at in future, but it's not something we've done.
Hi, I just had a quick question. Thank you. Really good talk. I really enjoyed it. I was just wondering if you've got reduced PV interneurons as well as reduced perineuronal nets, whether you've looked at the function of the PV interneurons or any sort of electrophysiology?
We haven't, but I would be really interested in doing that. I know there are people in Nottingham, so Tobias Bass's group. I can see some of the members in the audience would be interesting to work with them. Or likewise, I'm really happy to share more detail on the protocols and how we do the PCP, how we handle the PUPs and things to minimise rejection. And if others have the expertise to do that electrophysiology, I think it would be great to work together and characterise the model in more detail.
Also, just in terms of construct validity of the model, obviously we're not going to have in the real world neonates having PCP exposure. Are there sort of gene variants that disrupt NMDA receptors at that time point, or is there any sort of similar?
No, so that is, I will hold my hands up. So that is, in terms of the construct validity, there will be individuals who are exposed as a consequence of mum's recreational use. But no, we incorporated it because people were using it as a single hit, and it seemed logical, it seemed desirable. We did look, as I say, at maternal immune activation, because that's really nice in terms of the construct validity, and we have done some work on cannabinoid exposure as well. But yeah, I would say that is the odd thing. We do it because it works, but it doesn't necessarily. I mean, we're doing it because it causes excitatory inhibitory imbalance, and there will probably be other things going on in patients that are causing that. It's just it's not PCP. Anyway,
Thank you.
Thanks very much. I think probably move on.
So I'd like to introduce our next speaker, Dr. Rick Adams from University College London, titled Computational Modeling of EEG and fMRI in Schizophrenia Identifies Loss of Cortical Inhibition. So, Dr.
Thanks very much. Thanks for inviting me and organising. Okay. Okay, great. So I'm going to talk about trying to infer excitatory and inhibitory neuron function in vivo in patients with schizophrenia. First, may need to thank Elliot Hong and Dan Muthalon, who provided a lot of the data I'm going to talk about. So I'm sure most of you are familiar, but if not, I've tried to condense schizophrenia neurobiology onto one slide. Essentially, there's these two pathologies that we know most about. One is this idea of hyperactivity at striatal dopamine 2 receptors. We've known this for a long time because antipsychotics block these receptors. But a more recent hypothesis is this imbalance between excitation and inhibition that Maddie alluded to. And there's various kinds of evidence, genetic evidence, neuropathological evidence, pharmacological evidence that this may be the case. But the actual nature of this imbalance is a bit unclear. See, whether it's an NMDA receptor-mediated loss of function, whether it's due to some other reason, as Maddie was talking about, loss of perineural nets, for example, or whether it's concentrated on interneurons or pyramidal cells. It's a bit unclear. And so here we're just trying to explore what this imbalance might be. And so the options are perhaps a loss of pyramidal excitability. Now, in this excitability concept here, I've illustrated as an increase in this self-inhibitory connection, because that's what it comes down to in the model. But the model's not literally saying there's a self-inhibitory connection in these cells. All this means is a kind of loss of excitability in this population. So you'll see there's a lot, so this is why I'm introducing it here. Or there may be a loss of function of inhibitory interneurons, or there may be a loss of connectivity between these cell populations. And the data set that we were going to look at to try and explore this is data provided by Elliot Hong in Maryland of a reasonable size for neuroimaging data, more than 100 patients. And importantly, he administered multiple paradigms to the same patients. And they were very simple, straightforward paradigms. So resting state EEG, an auditory oddball task, a 40 hertz click train task, and some resting state fMRI. I'll describe them in a bit more detail in a moment. But the key questions that we wanted to look at were really, are there consistent changes in excitatory or inhibitory model parameters across all of these different paradigms in the same people? And do these changes relate to synaptic gain or excitability of these populations? And do they relate to symptoms of the disorder? And the approach that we were using is this approach called dynamic causal modeling. I'm not going to go into any of the details of it, but just to give a rough overview, the idea is that if you have a neuronal model of different cell populations connected up in different cortical areas and parameterized with things like excitability of those interneurons or excitability of those pyramidal cells, and then you ping it with some input then you can calculate how that neural activity will evolve over time. And if you can do that, then it's just a simple physics model will tell you how this activity can then be sensed in sensors outside the brain, either MEG or EEG. This is the model running in this forward direction here, producing observed data. What we really want to estimate are these parameters of the neuronal model. And so this is the clever estimation method that Carl Friston devised, this Bayesian approach that allows you to estimate individual subject level parameters for excitation and inhibition and connectivity and so on. And so we come up with a kind of cartoon model, for example, connecting temporal lobe to frontal lobe with forward connections and backward connections, and these excitatory, self-inhibition, excitability parameters that we're interested in here. But inside each of these cortical nodes is a microcircuit that reflects roughly the structure of cortical microcircuits in the brain. So we have superficial pyramidal cells, deep pyramidal cells, inhibitory interneurons, and the input spiny stellate cells. Obviously, the reality is that this circuitry is far more complicated, but this tries to capture the key elements. So let's go through these paradigms. So firstly, we have this resting state EEG paradigm. Here we're just taking a super simple approach and averaging the EEG all across cortex and looking at the power spectrum and trying to see if this changes in this microcircuit can reproduce observed changes in the power spectrum that we see. So what are those changes? So we see in the patients here in red, we see this small but significant increase in gamma power, in theta power, and a decrease in beta power in the power spectrum, eyes open or eyes closed. And so people have seen these changes before, and there's nothing new in Elliott's data here, but what we don't know is what might give rise to these spectral changes. So what we essentially did was just, instead of trying to fit the model, because the sources are not so clear in resting state EEG, we just tried to do some simulation to figure out what microcircuit changes might cause these spectral changes. So here we just propose perhaps all synapses are just a bit weaker in schizophrenia, or perhaps there's inhibitory interneuron loss of connectivity, or perhaps loss of inhibitory self-inhibition, so they're too overactive, or maybe inhibitory interneurons are underactive, or maybe pyramidal cells have lost excitability. And so to cut a long story short, we just simulated all of these scenarios and looked at the power spectra that were produced, and the only one that reproduced all the features that we saw was this loss of pyramidal excitability here that reproduced these data features that we were interested in. And then moving on to the mismatch negativity, this is probably the commonest EEG paradigm ever applied to schizophrenia investigations. It's a simple auditory oddball paradigm where you have a bunch of standard tones and then some rare deviant tones. And you compare the responses to the deviants to the responses to the standards. And we know the cortical circuitry implicated in this paradigm. People have done fMRI studies, and it involves auditory areas and a frontal node here at the top. But we're using the same microcircuit to model this, but now we're looking at the evoked response rather than the power spectrum. And if you plot this the usual way, you subtract the responses to the standards from the responses to the deviants. When you do that, you see this big negative deflection in controls around 200 milliseconds. But in the patients and in the relatives, this negative deflection is considerably reduced. This is the loss of the mismatch negativity. So we were asking what cortical connections or properties might underlie this change in the patients. And what we found is it's very much excitability of pyramidal cells in this frontal node that seem to underlie this difference between the deviant and the standard conditions in the patients. So again, loss of this pyramidal excitability. But Elliott, when he collected these data, he was interested in auditory symptoms like hallucinations or other auditory perceptual abnormalities. So he gave all of the participants quite a detailed auditory symptom scale. So he looked to see what model parameters correlated with symptoms in this task. And we expected it would just be loss of excitability, as we found in the main effect of group. But actually, this is what we saw on the right-hand side, but on the left-hand side, we saw actually a disinhibition seemed to correlate with auditory symptoms. And this was interesting because, of course, left IFG is essentially Broca's area, the area responsible for generation of speech. And so it seemed to be disinhibition here seemed to correlate with perception of voices and altered auditory function. In this third paradigm, a auditory click train of around 40 hertz is administered. And so you measure 40 hertz power in primary auditory cortex. And so this is what we're trying to analyze with our model. So why do people administer this click train? So the point is that the pyramidal cells and interneurons are connected in this very simple circuit, which naturally oscillates at around this 40 hertz gamma frequency. So the idea is if you ping the system at this frequency, then you'll set up endogenous oscillations at this frequency, and if there is something wrong with this circuit, then the power in that 40 hertz band will be reduced. And many people have used this paradigm in patients with schizophrenia before, and they have found what we found in Elliott's data, that in the controls you get this robust increase in 40 hertz power, but this is reduced in the patients and also reduced in the relatives, so it's not a medication effect. So then we wanted to ask which microcircuit parameters might change explain this change in power. So here we looked at both the relatives and patients combined versus controls, and then looked at the difference between the patients and the relatives, so the state of psychosis versus the genetic risk. And when we were looking at both patients and relatives together, we saw this kind of loss of input into inhibitory interneurons, but the difference between the patients and the relatives was this loss of excitability of pyramidal cells in bilateral auditory cortex. But we also went on to look at the correlations with symptoms. When Elliott originally wrote these data up, they noticed this kind of slightly counterintuitive relationship between the gamma power here on the x-axis and the symptoms, which was that although the patients seemed to show lower gamma power as a group, there was a positive correlation between gamma power and symptoms. So the patients that looked most like controls in their gamma power were the ones with the most auditory hallucinations, not necessarily the way round that you'd expect. And again, when we looked at the model parameters, we found that it's really disinhibition in this pyramidal to inhibitory interneuron circuit that seemed to correlate with auditory symptoms. Then lastly, looking at this fMRI paradigm, obviously in fMRI you don't have the temporal detail to be able to fit these microcircuit models, but you can look at kind of excitability in cortical areas. You just can't work out whether it's anything to do with pyramidal cells or interneurons. And when we analyzed the resting state fMRI data, we wanted to do it in a way that we could compare with the other paradigms. So instead of looking at connectivity of every area with every other area, which is what normally happens in resting state analysis, we just took a very focused approach and looked at this same circuit that we'd looked at in the auditory oddball paradigm, i.e. these auditory areas and the frontal node. And we thought it was reasonable to do this because if you're doing anything in a resting state fMRI scan, you're listening to a bunch of kind of unpredictable beeps and boops. So we thought it would be fair to look at activity in this circuit. And when we did that, to our surprise, we found exactly the same result that we found in the mismatch negativity EEG response. We saw this loss of excitability of these two frontal nodes on this auditory circuit in a completely different paradigm, the resting state fMRI. And so we also looked at symptom correlations and found actually the disinhibition in these kind of more auditory areas seem to correlate with symptoms, however. And so to put all this together, we saw these remarkably consistent results that across all of these 4 paradigms, there seemed to be this loss of excitability, especially of pyramidal cells, and especially in frontal areas in the patients as a main effect. So this is the case in the resting state, the mismatch, the 40 hertz, and in the resting state fMRI. But a striking thing that we hadn't really predicted was that, in fact, the symptoms of the disorder, the positive symptoms, the hallucinations, and other auditory abnormalities, didn't seem to relate to this loss of excitability. And if anything, they seemed to relate to disinhibition in A1, or in Broca's area, or in auditory areas more broadly. And so this suggested the really interesting hypothesis that perhaps symptoms might be the price that the brain pays for restoring excitability in brain networks. And what do I mean by that? So this was a hypothesis suggested by John Crystal in a paper in Biological Psychiatry some time ago. He kind of proposed that maybe the fundamental problem in schizophrenia is this glutamatergic synaptic dysfunction. And then you get this, what he called allostatic adaptation, this kind of downregulation of inhibition in order to preserve the firing rate of pyramidal cells. And then this, he then proposed, cause its own problems, and you get this kind of sequence of events leading to the disorder. And so it may be that you see this kind of primary loss of excitability and then this downregulation of interneurons that tries to compensate. Alternatively, of course, you could have subgroups within the disorder, some with excitatory dysfunction, some with inhibitory dysfunction. And we may be seeing a kind of merge of these two things. It's hard to tell exactly from these data. But if this theory is correct, one should see these same changes and relationships that we'd seen in chronic illness, in Elliott's data, in the prodrome. So we went to look at a data set collected by Dan Mathalon in UCSF, the biggest prodromal data set so far collected in schizophrenia research. And he took hundreds and hundreds of patients with prodromal symptoms and then measured their EEGs, and then 2 years later gauged whether they had converted to full-blown psychosis or remitted. And so he had 90 people who remitted and 70-odd people who converted. And here we're looking at a mismatched paradigm again, and the same paradigm but involving a kind of active target. So you have to make a response to the target rather than just listen passively. And when we compare these high-risk subjects, the remitters versus the converters, in both cases you see this. In the converters, in light green here, you see a smaller mismatch negativity response, and you see a smaller P300 response. So my PhD student, Julia, modeled these data. We used the same model that we used before for the mismatch negativity, a slightly different one for the P300 because this active element requires this dorsal attention network. But we're also using this same microcircuit model and looking at pyramidal cells and interneuron excitability. And sure enough, Julia found that in both the passive and the active oddball conditions, it was a loss of pyramidal excitability that was the difference between the converters and the remitters in both cases. But when she looked at the relationships of positive symptoms with the model parameters, again, she saw this disinhibition of pyramidal cells in the mismatch negativity and in the P300 paradigm that seem to relate to symptoms. So to summarize that all, we see this quite consistent relationship between loss of pyramidal excitability and schizophrenia, but this relationship of loss of inhibitory interneuron function with positive symptoms, this might be due to this downregulation that John Crystal proposed, or it could be due to presence of subgroups, some causing primary interneuron pathology, some perhaps more genetically mediated pyramidal cell pathology. And we've seen this in both chronic illness and in the prodrome. And so the outstanding questions really are whether we can use these methods to try to stratify prodromal or first episode patients who might benefit from glutamatergic treatments, because obviously these treatments have failed to get through phase 3 trials so far, but perhaps with some stratification based on these methods, they would achieve that. And then secondly, if there are subgroups within the psychosis spectrum, we might be able to identify them this way and give different treatments to different groups, and that will be our hope. Thanks very much.
Fantastic talk, Rick. I don't know whether this is actually on. Hello? Hello? Oh, there we go, sorry. So we're just looking for some questions. We're a little bit ahead of schedule, so there's plenty of time to quiz Rick on everything. We have one just here.
Hi, thank you very much for that. Sorry, this is a bit of an esoteric question, but I was just wondering, with the ASSR, if you looked, in addition to the auditory cortex, at prefrontal or other regions, and whether or not you saw any discrepancies in the responses.
So in these data, no, because they were the EEG, and we didn't feel super confident about localizing sources beyond the primary auditory cortex. But in some of Peter Ulhaus's MEG data that he's lent to us, but I haven't presented here, I mean, when he source localized that paradigm, he found a kind of hippocampal source and a thalamic source, but we haven't finished that analysis, so I couldn't tell you what we found. But are you thinking there may be some difference between frontal responses in that subgroups or something in that paradigm?
Well, it's a question. I don't know the answer.
Oh, okay, all right.
Have we got another question for Rick? Here at the front, please.
Hi, Rick.
Hey, Nick.
I was curious about just to hear a little bit of more information about the correlations that you said you observed with the symptoms.
Mm-hmm.
I guess especially as far as the excitatory abnormalities go. You mentioned out loud positive symptoms. Are we talking PANS, and is it specifically positive symptoms that you saw, or something more across the board?
Yeah, so in the Naples data, the prodromal data set, they collected this SOPS questionnaire, which is basically like a positive symptom questionnaire, but slightly more sensitive than a typical PANS, because obviously these people don't have quite as powerful symptoms as people with schizophrenia do. In Elliot's case, he collected a very specific auditory symptoms questionnaire, so it wasn't an overall positive symptoms measure. It was very focused on auditory symptoms, and it was much more sensitive. So it asked things like, do things sound a bit louder than they should do? So things like this. And we only really looked at that in connection to the EEG paradigms. But we did look at some PANS measures with the resting state fMRI and found actually similar positive symptoms as a whole. Even if you regressed out the auditory ones, you still saw this relationship of positive symptoms to disinhibition. But because it was only in that one paradigm, I didn't make a big thing of it. But we did see some relationship beyond the auditory, yeah.
Great, thanks, Rick.
So we've got another question here over on the left-hand side just here. Thank you.
Hi, thanks. I really enjoyed that talk, really beautiful design. I was just wondering whether you had any data on cognitive symptoms, and particularly sort of with the gamma data, whether it correlated with positive symptoms.
Yes,
cognitive symptoms.
So I didn't show it, but there was a single kind of processing speed cognitive measure that Elliot had collected. We haven't looked in the Naples data, but we did actually see across 3 of those paradigms, there was a relationship of pyramidal cell excitability to cognitive performance, but it was only consistently there in the controls. In the patients, that seemed to be lost. It was only present in 1 out of 3 paradigms. So really, it seemed to indicate the better your, I mean, this is extremely speculative, but the better your NMDA receptors work, or the more you can sustain activity in your pyramidal cells over time, the better your processing speed measure was. But because that relationship, just seemed to be lost in the patients, we didn't, I didn't mention it here, but there did seem to be a relationship, even in A1.
Wow, interesting.
Yeah.
Also just wondering whether you had any genomic data from the patients that would enable you to match gene variants with any of this, or no?
I mean, he did collect it, but I was too scared to try and analyse that.
So we've probably got time for maybe one more question, if anybody has anything. No? Okay, well,
Cool.
thank you ever
Thanks
so
very much.
So hopefully we have our third speaker online, Professor Oliver Howes. There he is. So I'll hand over to Professor Oliver Howes now. Thank you.
Thank you very much, and thank you for putting this session together. Sorry I can't be there in person. So I'm going to be talking about corticostriatal circuits linked to psychosis and back translation into a rodent model. Here are my disclosures. The only thing that's relevant actually to what I am going to be talking about is that I'm going to be talking about the effect of a novel compound that was developed by Synovian, and they were involved in that study. And I'd like to thank lots of people who've helped with the studies that I'm going to talk about. You see their names here, and there's a picture of us at Christmas. But also, I really want to thank the patients and volunteers who've taken part in our studies. So this is the outline of my talk, and I'm going to talk to begin with just about the dopamine changes in schizophrenia and where the textbooks seem to be wrong, what we can do about targeting this and the link to cortical function and where we might get better treatments in the future. So what do the textbooks all say about the dopaminergic changes that underlie schizophrenia? Well, they all say it's mesolimbic, and overactivity in the mesolimbic projections underlies the positive symptoms in particular. That's been around in textbooks for decades. But developments in PET scanners recently have meant that we've been able to actually look at this question of whether the major dopamine change really is in the limbic parts, limbic projections. So this is a slide that summarizes the topographical organization of projections to the striatum, and track tracing studies in non-human primates and other species have shown that you have this particular topography. So you have these projections from the ventral tegmental area to the nucleus accumbens and ventral striatum, which we term the limbic part of the striatum. And this also receives projections from amygdala and other limbic areas in the brain. Then more dorsal striatum, you get projections from different areas of the substantia nigra. And these receive projections from prefrontal cortex. So this is the head of the caudate and part of the putamen. And so is often termed the associative striatum because it's linked to associative cortical regions. And then more dorsally still, you see the part of the striatum that receives projections from motor and sensory motor and sensory cortex. And that's often termed the sensory motor striatum. So with these higher resolution PET cameras that we've had in the last decade or so, we've been able to look at these areas within the striatum. And something very interesting has emerged from all that. There have been a number of studies now, and we meta-analyzed these a couple of years ago. And here you can see that actually, where you look at the case control differences in dopamine synthesis or release in the striatum, the big changes are not in the limbic area, shown in red, but actually in the associative part of the striatum, in blue there, and to a lesser degree, but still quite large, in the sensory motor part of the striatum, in yellow there. And these are pretty large effect sizes, 0.7 to 0.8 for the associative striatum. So quite a big increase in dopamine synthesis and release capacity in schizophrenia. And this has now been seen across multiple studies around the world since our first one showed this. So this is very interesting. It tells us that this mesolimbic theory is probably not correct, and it pinpoints connections to the frontal cortex as being particularly important. More recently, people have started to look at another part of the striatum, and the part of the striatum that's projecting to, part of the midbrain that is projecting to the associative part of the striatum. And we're able to look at this in patients using a different imaging technique, a technique that measures neuromelanin. And this is neuromelanin is essentially what happens when dopamine gets old and dies. It's dopamine conjugated with iron and some other compounds, and essentially as a long-term storage. And of course, this is what it appears black in the substantia nigra, and why the substantia nigra is termed the substantia nigra. People have started measuring this in schizophrenia. So this is a marker of the long-term overactivity in the dopamine system. And here you see some data from a study that we've just done, where we've looked at this neuromelanin signal in the midbrain and compared it in people with schizophrenia with healthy controls. And you can see in green the areas where it is elevated in people with schizophrenia. Now, this is nothing new. A number of other studies have shown this. This is pretty reliably seen now, that people with schizophrenia show this increase in neuromelanin signal in the midbrain, telling us that not only is there evidence for striatal overactivity, but also evidence in the midbrain. But what we did here that was interesting was we looked within the midbrain at the voxel level where the changes were most marked. And you can see the color coding shows you the deeper green. It shows you the areas where there's the most significant elevation in people with schizophrenia. And this turns out to be in the dorsomedial part of the substantia nigra. And that's very interesting because that is the part that, of course, projects to the associative striatum. And Luke, who did this study, also went on to look at the relationship between this midbrain neuromelanin measure and striatal dopamine synthesis capacity. And he acquired the two measures in the same patients and showed that they were both quite strongly related, as you can see here. So if you start putting this together, we're seeing that there's overactivity in this particular part of the circuit, projecting from the dorsomedial substantia nigra to the associative striatum. This, of course, raises the question about where the prefrontal cortex fits into all this. Now, we and others have been looking at the relationships between prefrontal cortical activity and various other markers in vivo in patients, and have shown that these are related to this dopaminergic change in the striatum and other markers of striatal activity. So they do seem to be related. But we were particularly interested in what part of that circuit is maybe driving the striatal changes. So we know that there are projections from the prefrontal cortex, glutamatergic projections down to the midbrain from tracing studies. This is a photomicrograph showing glutamatergic projection synapsing onto Th positive neurons in the midbrain. And as you've already heard from the previous talks, one of the most prevalent current theories of schizophrenia is that it starts with an imbalance in this coordination between GABAergic and glutamatergic signaling, with an excitatory inhibitory imbalance in the cortex. And we wanted to test whether this could lead to overactivity, the midbrain dopaminergic projections to the striatum. We tested this in an animal model. We used special K. Don't worry if you had special K for breakfast, because this is actually the street name for ketamine. And the reason we focused on this is because we'd already looked in a study of long-term ketamine users who had taken it repeatedly, but then become abstinent, been abstinent for a number of years. And what this study showed is that they continued to experience psychotic, low-level psychotic symptoms. So here you can see the level of these symptoms on the vertical axis, ketamine users, ex-ketamine users here in red, still several years later experiencing these symptoms. Ketamine, of course, it's an NMDA receptor antagonist, as is PCP, and you heard about that in Maddie's model. But this was particularly interesting to us because it not only produces psychotic and negative symptoms when it's given acutely, but if you take it repeatedly, at least in these ex-ketamine users, you seem to get this persistent psychotic syndrome. So we then wanted to test this in mice and the effect on the dopamine system to answer this question. And we gave ketamine repeatedly to the mice over 5 days. Then they were washed out of ketamine over 2 days, and then they had a PET scan with the same PET measure that we use in patients. And here we found that indeed this ketamine model resulted in increased dopamine synthesis capacity in the striatum. And remember, this is something that is not when that ketamine is actually on board in the animals, but after they've been washed out of it for several days. So that's interesting. It tells us that ketamine could be acting on this system, but it doesn't tell us where in the circuit. So we then went on to use DREADS to express an inhibitory receptor, specifically in the midbrain dopaminergic neurons. And here you can see the co-localization with the TH marker for dopamine neurons in green, and M-cherry showing the expression of the viral construct for the inhibitory receptor. And you can see the co-expression down there. And we were able to label a large proportion of the dopamine neurons in the midbrain, and importantly, negligible labeling in non-dopaminergic neurons, as you can see on the bar chart on the right. We did some electrophysiology of these neurons, and here you can see a recording from dopamine neurons in these animals, and you can see them the dopamine neurons firing nicely. But then when we gave CNO, which activates this inhibitory receptor, you can see this silences the dopamine neuron firing. So this allowed us to selectively silence the midbrain dopamine neurons. So we used this to test whether we could block ketamine's effect. So here you can see again dopamine synthesis in the striatum on the vertical axis, the saline controls, saline with the CNO having no significant effect there. Ketamine, again on its own, reproducing this effect, elevation in striatal dopamine synthesis in the animals. But when you use the CNO to block those midbrain dopamine neurons, you can see that this is reversing that effect or blocking that effect, and it's back down to control levels. So it seems that this ketamine effect does require midbrain dopamine neurons, and that of course suggests that maybe this could be a target that we could use to develop new treatments for patients. And one receptor that is expressed in dopamine neurons that's very interesting from this respect is the traceamine receptor, the TAR1 receptor. So this is expressed in dopamine neurons, and it regulates their activity. It's part of the autoregulatory mechanism. Activating it reduces dopamine neuron firing. And we worked with Synovian, who developed a selective compound for this to test this. Firstly, Alice Petty here did some fast scan cyclic voltammetry together with Stephanie Cragg's group, to test the effect of this drug, this TAAR1 agonist, showing that the TAAR1 agonist reduced dopamine neuron firing from striatal slices. So you can see here in blue this dose-dependent effect of ulotaront, the TAAR1 agonist. So it's working to reduce dopamine neuron activity. We then wanted to test whether it could block this effect of ketamine. So again, we used the same subchronic ketamine model. And then just before they had the scan, the animals received a dose of this TAAR1 agonist and then the PET scan after the ketamine had been washed out again. And here you can see the effect. So again, vertical axis dopamine synthesis capacity, the two control groups, CEP or ulotaront, the TAAR1 agonist on its own, had negligible effect on dopamine synthesis capacity. Ketamine again increased it, but when you've given the TAAR1 agonist at the same time, it's blocked the effect. So it seems that that we were able to block the effect of ketamine with activating TAAR1 receptors with this agonist. We then went on to test this in schizophrenia patients. So this is a proof of concept study. The patients with schizophrenia were given ulotaront, added on to antipsychotic. These were patients with ongoing symptoms, and they had the TAAR1 agonist for 2 weeks, and then they had the PET scan repeated to see if this reduced dopamine synthesis capacity. And indeed, this is what we found. So there you can see across all the striatal subdivisions, actually, it reduced dopamine synthesis capacity. And here is the voxel-wise map of the effects. So interestingly, the most significant effect was seen in the head of caudate, and you can see in the darker red the degree of significance, but it is seen across the striatum. And we looked at the relationship between this change and dopamine synthesis capacity. And what we see is that greater reduction in dopamine synthesis capacity was associated with a larger improvement in positive symptoms, but not in other symptoms, not in negative or total symptoms. So it seems specific to psychotic symptoms. Now, how does this link to the cortex? Well, Michelle in her ketamine model also went on to use DREADS to manipulate parvalbumin positive interneurons, because we think that ketamine is acting to inhibit the firing of these parvalbumin positive interneurons here in blue, dysregulating those glutamatergic projections to the midbrain. So she theorized that if we activated those parvalbumin interneurons, perhaps we could block ketamine's effect. So again, this time expressing the excitatory receptor specifically in parvalbumin positive neurons. And here you can see us recording from these parvalbumin positive neurons and applying CNO increased their firing. And she then tested their effect on dopamine synthesis capacity. And you can see that activating those parvalbumin positive interneurons with CNO blocked the effect of ketamine on dopamine synthesis capacity. And remember, these parvalbumin positive interneurons, it was only those in the cortex that had been controlled or infected with the DREAD. So it seems that activating the parvalbumin positive interneurons blocks ketamine's effect and is consistent with this model of EI imbalance leading to the dopaminergic changes. Of course, this raises the question, what's going on in schizophrenia? Ketamine is just a model. We think actually it's to do with the balance of synapses on these GABAergic and glutamatergic projections, and there is some emerging data on that, from studies in patients that we're currently looking at, and that's a theory that we've published on last year as well. So just to summarize, higher dopamine synthesis capacity is specifically or most markedly seen in the associative striatum, and other studies have shown this is linked to the psychosis. Now we're seeing that higher neuromelanin accumulation seems to be most marked in the dorsomedial substantia nigra that projects to the associative striatum. This subchronic ketamine model induces striatal dopamine synthesis capacity elevations seems to require the midbrain dopamine neuron firing and can be blocked by cortical parvalbumin neuron activation or a TAAR1 agonist drug. And this TAAR1 agonism seems to reduce striatal dopamine synthesis capacity in patients with schizophrenia. Thank you for your attention.
Do we have any questions for Oliver in the room? Yeah, just at the back.
Hi, thanks for the talk. You gave the, was it mice or rats, a 30 milligram per kg dose of ketamine, and I was just wondering how that's translationally relevant.
So this is, it's a subanesthetic dose. It's equivalent to the doses that have commonly been used in the acute ketamine challenges looking at the induction of symptoms. But of course, this is much higher than patients, people commonly abuse and take ketamine. And this is what we've seen as acutely induces symptoms when you translate it into what the human equivalent would be.
Hi Oliver, nice talk. I was just wondering regarding the ketamine model that you have been using. So do you think that the NMDA receptor dependent effect of ketamine is playing a role in this? And if so, do you think that the effects can be studied in a different model? And your thoughts on that?
Yes, yeah, I think it's a great question. We think that it is via NMDA receptor actions on these inhibitory interneurons, but of course we don't know that, and we haven't tested that in this model. With this model and with other models, with PCP and other ketamine models as well, people have shown that you do get loss of these parvalbumin levels in prefrontal cortex. So as you see in schizophrenia as well in post-mortem. So it is reproducing this aspect of the pathophysiology and does seem to be impacting on these parvalbumin interneurons. But whether it's via NMDA receptors, we don't know.
Hi, Oliver. Great talk. I was just wondering with the, we know that ketamine disrupts working memory, and whether when you increased sort of parvalbumin activation, whether you were able to look at any effects on cognitive symptoms.
Sadly, we didn't. We weren't able to do that. It'd be a great thing to do.
Hello, Oliver. Lovely talk. I wanted to ask you about the MTAR, the TAR agonists. So in this case, you show that so this is an acute exposure to the drug?
That's right.
So is there any evidence that long-term administration of the drug, and how does that affect dopamine levels? The reason why I'm asking that is in terms of dual diagnosis patients with cannabis, for example, there's a suppression of dopamine, which also correlates with higher positive symptoms. So how does that relate? Effectivity.
Yes, that's a good question. And so in the animals, we're hoping to go on to test the long-term effect. We haven't tested it yet. Of course, in the patients, they received the TAAR1 agonist for 2 weeks. So we didn't see any evidence of tachyphylaxis in there. They showed a reduction in dopamine synthesis capacity. So in over 2 weeks at least, the TAAR1 agonist is seeming to have an effect. In terms of dual diagnosis, as you say, they seem that they show this blunting of effect, but still a relationship with the induction of psychotic symptoms. TAAR1 agonist is probably a better strategy than a D2 blocker for these people, because this still permits normal dopaminergic signaling. So it's a homeostatic, more of a homeostatic mechanism, I think, than a D2 blocker, which is like hitting the dopamine system with a sledgehammer. So it hasn't been tested, to my knowledge, in dual diagnosis patients, but I think it would be interesting to do that for that reason.
Are there any more questions? No? Okay. So we'll move on to our final. Thank you, Oliver.
Thank you.
We'll move on to our final speaker, Dr. Christian Wood from Cambridge.
Hello, everyone. Just checking that everything works up here. So yeah, I'm going to be talking about some work that we've been doing in Cambridge, trying to understand specific circuits and how they relate to aspects of symptoms with regards to mood and anxiety disorders, specifically relating to this kind of loss of reward, which is associated with anhedonia, and then aspects related to responding to threats that we akin to anxiety. And in a non-human primate, so specifically this is the common marmoset. This is a New World monkey that we utilize in Cambridge. So I'm a postdoc working with someone called Professor Angela Roberts, who's been kind of pioneering marmoset research for the past 30 years. And most importantly, obviously, thank you to the BAP for putting on this symposium. So, why are we kind of trying to understand the neurobiology of mood and anxiety disorders? Well, I mean, I imagine most of the people in this room are fairly aware of the fact that they are incredibly prevalent globally. Up to around about 500 million people worldwide suffer with these disorders, of which it's around up to about a third of people aren't effectively treated. And you could imagine the kind of personal, social, and economic burden that kind of causes. And what feeds into this to a certain extent is that we have these kind of first-line treatments as well as psychological treatments. And there's a lot of heterogeneity in terms of the different types of symptoms that people might experience. So here on the right-hand side, you have the DSM-5 criteria specifically for depression. And you could have an individual in red and an individual in blue having essentially completely different symptom profiles, but still have the same diagnosis. And as you can imagine, with this heterogeneity in terms of symptoms, you also then have heterogeneity in terms of the neurobiological dysfunction that's observed across these different patient cohorts. And that obviously is quite difficult to kind of marry up with the fact that we only have certain single-line treatments for these disorders. And when I'm speaking to the idea of this neurobiological dysfunction, in terms of in humans and in the kind of main powerhouse of studying these kind of cognitive and emotional functions in the prefrontal cortex, here we have the human brain, and in red is every region that has kind of been reported in one study or another that is shown to be dysfunctional. As you can imagine, that is most of the prefrontal cortex to a certain extent. And what's even more alarming is the fact that in terms of the different sets of patients or studies that you look at, you might see a hyperactivity or a hypoactivity, depending on the study and the subset of patients you look at. And really, from a basic science perspective, as a researcher trying to understand prefrontal function, there is an element that we aren't entirely sure how these individual regions really map onto different aspects of cognitive and emotional functioning, and perhaps when they're dysfunctional might also feed into the symptoms of, say, mood and anxiety disorders. And really that's where I'm coming from, trying to understand the different parts of the brain up in the prefrontal cortex in our marmosets, and how they actually relate to these different processes. Now, why are we studying prefrontal cortex in a non-human primate? And I mean, that's most important when you think about the actual architecture of the brain across different species. And hopefully you can see on the left the human brain and on the right the rat brain. And without going into too much anatomical detail, you can probably see in terms of the colors there's quite a big difference. And obviously rodent studies, as has been alluded to in the work of the speakers before me, there's a lot of work in the cortical function of rodents. It's incredibly useful in terms of understanding psychiatric disorders. When it comes to thinking about lots of aspects of prefrontal function, it's about utilizing a species, in my mind, that more marries onto the homology that you see in humans. So here in the middle you have a marmoset, this is the medial view of the brain across the three species, that so very well matches what you see in a human, of which the human and the marmoset have, say, between 60 to 80% of their brain given over to cortex versus, say, around about 30% in a rat. So I'm here to talk about the subgenual cingulate cortex. And what do I mean when I say this subgenual cingulate for the uninitiated? Well, it's part of this subgenual region below the corpus callosum, under the knee or genu of the corpus callosum, that often was also referred to as part of the ventromedial prefrontal cortex, or VMPFC. Now, I hope you can see I'm specifically going to be talking about area 25, but this gamut of regions called the subgenual cingulate is actually lots of different regions kind of combined together. So that often is a bit of a problem when it comes to neuroimaging studies, thinking of the correlational activity of these structures, in the fact that what gets called area 25 might not necessarily architectonically be actually area 25. And this is kind of a host of studies in control individuals showing that they have a heightened activity in area 25 just in normal individuals in a SAD situation versus, say, a landmark study by Helen Mayberg looking at deep brain stimulation in treatment-resistant depressed patients, showing this kind of hyperactivity in a region labeled singular area 25 that is actually ameliorated following deep brain stimulation. But also Helen Mayberg has kind of shown with regards to amelioration of this hyperactivity, SSRIs do a similar sort of process as well. And then obviously we have also a study that's perhaps more relevant to us in our recent history, following a vaccination and neuroinflammation, kind of negative mood changes associated with that were directly correlated with increased activity in, again, this region-associated area 25. But hopefully, as I've kind of alluded to, this brain region, what we call area 25, is floating around a little bit in this ventromedial and subcallosal box that I have here on the right. And really the issue for us as basic scientists is wanting to know, well, what is the causal function of this region? And can I necessarily really relate it to this correlational finding in humans? And that's kind of why we wanted to tap into exactly what happens when we overactivate this region in our marmosets in area 25. So to do that, we did a very basic study in its first instance, and that is by pharmacologically overactivating the brain region. And this is through placing indwelling cannula directly above area 25 so we can infuse a drug called dihydrocanic acid, DHK, to reduce glutamate reuptake in the region to essentially overactivate area 25. And then we can look at kind of the emotional responses associated with that. And the way in which we kind of study these responses, it's not just in terms of, as a behavioral neuroscientist, not just looking at their behavior through video recordings, but also we looked at their cardiovascular responses as well, because that's an important component of the emotional response. And to do this, we use wireless cardiovascular telemetry. So we place a telemeter into the descending aorta of the marmosets in a one-off surgery so that we can actually study their blood pressure and heart rate whenever we're conducting our testing. So speaking to the ideas of these different components I'm wanting to look at with regards to area 25, we looked at a lot of different paradigms over the course of the research that we've been doing, but the two that I'm kind of primarily going to be talking about is the first is about reward anticipation. So this is a part of this diminished aspect of pleasure that is observed in depressed patients, or anhedonia as a core symptom. And the blunting of anticipation is kind of seen in individuals with depression, but there's lots of other factors that kind of feed into this anhedonia. And in terms of looking at reward anticipation in the marmosets, we do this with a very basic task. It's called appetitive Pavlovian conditioning, and I'm sure we're all probably fairly familiar with Pavlovian conditioning. But for the marmosets, it's a case that they have two sounds that are paired with two outcomes. One 20-second tone gets paired with the fact they're going to get access to that absolute favorite food in the world, which is marshmallows, like you have in a coffee shop. And then there's another sound that gets paired with access to no food. And what these animals understandably quite rightly do is develop a Pavlovian response in anticipation of getting access to the marshmallows. So you see a nice blood pressure increase here in orange versus the CS minus, which predicts no access to food. So a beautiful discrimination they display. And what we do after we've overactivated area 25 is you actually see this reward anticipation being blunted. So they're no longer able to really show this reward anticipation. And this here is kind of the blood pressure change from cardiovascular response, but it's also the case for their behavioral response, a kind of anticipatory head jerk movement. But importantly, in activating 25, it's not a case that we're completely blunting them emotionally, because when we look in a more of a threatening condition, we see something different. Now, the way in which we kind of look in a more negative context is by placing them in the top right quadrant of their home cage and then introduce them to a novel human, which For a marmoset as a prey species, a novel human might be there. It's a case of, are you friend or are you foe? You may be there to give them marshmallow as a friend, or it may be that you're picking them up to give them some sort of husbandry procedure. So what happens is a novel person stands in front of the cage and maintains eye contact with them for 2 minutes. So it's kind of this uncertain threat, almost akin to what would be an anxiety response. We look at a range of different behaviors that are combined together to a threat score. And what happens is when you overactivate area 25, you actually see this heightening of this threat response in comparison to, say, a controlled infusion such as saline. So, the fact is when we overactivate area 25 pharmacologically, we're really inducing this negative affective state. And the idea that you're blunting reward akin to kind of an anhedonia-like phenotype, but then also heightening responses into more uncertain threats or ambiguous situations. So knowing we have these very clear behavioral effects through activating this region that we know is associated with depression, area 25 in our marmosets, we really wanted to study, well, what are the networks that are kind of being, that are contributing to these effects? And the way in which we did that is by doing 18-fluorodeoxyglucose PET imaging. So fluorodeoxyglucose is essentially glucose that isn't utilized by cells particularly well, and it's a marker for network activity, such that you can overactivate area 25, administer this fluorodeoxyglucose that's radioactive, and then put them in either a rewarding situation or a threatening situation for half an hour, subsequently anesthetize them, and then look at all the different brain regions that have been more active, that have taken up more FDG, and then look at the actual overall network that's impacted by activating area 25. And that's kind of summarized in this figure here. So when you overactivate area 25 in a rewarding context, this appetitive Pavlovian paradigm, you can see that we get an increase in activity in a mildly dorsal anterior cingulate and dorsal medial prefrontal region. We get increased activity in the anterior insula, as well as then a decrease in activity in a brainstem region, that includes the raphe nucleus. But in terms, we see a very different thing when we look in a threatening context, in the fact that we see when we overactivate area 25, an increase in activity in basomedial and basolateral portion of the amygdala, as well as, again, a little bit in the insula, but then we see an increase in activity in the hypothalamus. But then really interesting to us as well is that we see a decrease in the left side of the dorsolateral prefrontal area 46. So this kind of more higher order top-down control region is being suppressed in a threatening context when we overactivate area 25. So after kind of seeing this very clear difference in terms of the different networks in these different situations when you overactivate area 25 that may contribute to these behavioral outputs, we wanted to actually ask the question of, well, what specific circuits might actually be associated with driving these systems? And the way in which we wanted to do that is utilizing chemogenetics. But before we start on the chemogenetic front, we really need to understand anatomically what are the regions that actually project from area 25, and where are the terminals from area 25 neurons going down to. And our first instance kind of fits with the theme for our symposium today, is the part of the limbic striatum that Oliver quite beautifully explained to us. But area 25 has beautiful projections down into the nucleus accumbens. Hopefully you can see from this anterograde fiber tracing study that we've been doing in our marmosets. Lots of green fibers down in the nucleus accumbens. But then hopefully you can also see on the right-hand side some work by Suzanne Haber in the macaque monkey showing a very similar profile in terms of the connectivity of area 25 to the nucleus accumbens as well. So, that's one of our downstream pathways we wanted to look at. But the second kind of comes from our own work, excuse me, knowing that activating area 25 was associated with increasing activity in this basolateral portion of the amygdala, of which we know, importantly, that the amygdala is not just involved in threat-related responses, but also reward-related responses too. And importantly as well, the same could be said for the nucleus accumbens. So really, we weren't sure whether one or other was be associated with this blunting of reward anticipation or heightening of threat response that I've kind of referred to before. So as I mentioned, we kind of was conducting a chemogenetic study, and the way in which we did that is by placing the M3 designer receptor, excitatory designer receptor, into a virus under a CAMK2A promoter. So this primarily has these M3 excitatory receptors expressed in excitatory projection neurons. such that when we give the designer drug of this dread, systemically, you can activate all of the projections of Area 25 and study the effects of that. But for us, thinking about the actual projections we wanted to study, we essentially cannulated again these downstream structures of the nucleus accumbens core and basolateral amygdala, such that we can then infuse these designer drugs, so either clozapine N oxide or the more novel compound, which is quite a bit more potent, that using far less drug, deschloroclozapine or DCZ, down into these terminal regions such that you can activate the neurons between Area 25 and, say, the accumbens or the amygdala. So as a good proof of principle for us, we were like, well, what happens when we activate all of the projections out of Area 25? Do we see a similar response to when we pharmacologically activate the brain region? And the answer is yes. We replicate beautifully the blunting of reward anticipation. So as when we give a systemic dose of CNO, but we've seen the same with DCZ, we see a blunting of reward anticipation. So this is this blood pressure response to this sound that predicts access to reward, the CS+. And what we see is by giving higher doses of CNO, you see this blunting of this reward anticipation. And then importantly, you also see this heightening of this response to this novel human, in this human intruder test, as indicated by this overall threat score that kind of combines all the different measures together. And for those of you wondering, it's important to point out that these cardiovascular and behavioral effects are not present without the dread there. So we've conducted everything here without the dread virus, and there is no effect. It's primarily dread mediated in our minds, given the fact that after we give the virus, we then see these effects. So speaking to the data and manipulating the individual projections, so from the aspects of reward anticipation in this Pavlovian conditioning paradigm, by infusing CNO either into the accumbens or the amygdala, we then looked at the blood pressure changes. So if we first look at the control data, so we have the CS- and the CS+ here when we infuse vehicle into the accumbens in this kind of blue section on the left. You can see the nice discrimination that happens as a beautiful control. But when we infuse CNO down into the nucleus accumbens, we're getting this cardiovascular blunting here on the left. So yeah, hopefully you can see the difference between this gray bar and this blue bar. And then we're seeing the same for behaviorally in terms of this gray bar here. They're showing these anticipatory behaviors to the CS+ that's then diminished when you infuse CNO into the accumbens and activate the 25 to accumbens pathway. But importantly, this is not, it's not just a case of, it's all the pathways do this. When we infuse CNO down into the amygdala and activate the 25 to amygdala pathway, you can see that there's no effect. So it seems to be that this nucleus accumbens projection is really mediating this blunting of reward anticipation. But then it's a case of thinking about the threat context. The nucleus accumbens projection isn't the core one for that, in the sense that it doesn't impact everything, because when we look at the threat score in this human intruder test, when we infuse CNO into the accumbens and activate that pathway, you can see that there's no difference between vehicle and CNO. But importantly, we have a bit of a dissociation happening in the sense that when we activate the area 25 to amygdala pathway, we're then getting this driving up of this threat response in comparison to the vehicle infusion. So in our minds, this is a bit of a double dissociation between the area 25 pathways of the accumbens and the amygdala that are kind of driving these two different behavioral correlates associated with depression that we're seeing when we manipulate and overactivate area 25. So just kind of to give you a bit of a summary, area 25, as we know, when we causally overactivate the brain region, seems to induce this really negative affective state. And I've not necessarily spoke to other aspects that we've looked at in terms of the cardiovascular function at basal levels and aspects of fear-related processing that's going on with area 25, or even endocrine changes that are kind of observed in that Nature Comms paper. But really, it's important to kind of point out that there are very clear, distinct reward and threat-related circuits being driven by this activation of the region. And the fact is that it's driven by different projections from area 25, of which the blunting of reward anticipation is driven by a pathway to the nucleus accumbens, and then threat responsivity is being driven by this pathway to the amygdala. Now, after that whistle-stop tour, there's a lot still to be answered in terms of the function of area 25 and all of its downstream connections. But on top of that, it's also the case that what exactly might actually be controlling area 25 itself in terms of a cortico-cortical connection. Now we have this is where we've kind of been spending the last 2 years thinking about in terms of data. And this kind of stems from this work where we found this decrease in activity in left dorsolateral prefrontal area 46 in a threatening condition when we've overactivated area 25, implying there's some sort of either direct or indirect connection between the two regions. And we're hoping to have some data out in the next few months about this. This is kind of a lot of work that's been done to try and utilize these chemogenetic strategies in primates, especially when thinking about intercortical connections. But furthermore, when thinking about area 25 and kind of depression as a whole, there's a case that depression often onsets during adolescence. And it's a case that there may be that there's a particular vulnerability during that period as it relates to either less top-down control or bottom-up kind of influence to area 25 with data here from Steve Saviak in the marmoset showing that there's quite an extended developmental timeline for area 25 over the course of adolescence going into adulthood at about 24 months. And it's really a PhD student and BNA scholar, Spatika Jayaram, who's here, who's kind of pioneering this with a postdoc in the lab, Dr. Taylor Lynn Jones, trying to understand at what time point does area 25 come online, if we can activate the brain region at certain distinct milestones across adolescence, whether this kind of disruption in terms of reward processing is observed or heightening of threat responses. And then finally, I've been quite lucky in getting a bit of money from the Brain and Behavior Research Foundation to kind of study more chronically what it is that area 25 is necessarily contributing in our marmosets. So obviously a lot of the data I've talked to here is what happens when you acutely activate, but as you can imagine from a clinical perspective, it is often more chronically active over time, and that's obviously what imparts the range of different symptoms perhaps that you would observe in individuals that show this increased activity. And really, you know, I'm also hoping to look not just at behavioral aspects, but if you remember that symptom list that I showed you on the right-hand side, there are a lot of physiological aspects that are still also going on related to, say, fatigue and arousal levels, to sleep and disordered sleep and insomnia, but also aspects of endocrine function on top of that, or inflammatory markers too. So that's kind of where I'm starting to look now on top of, as well as thinking about area 25 and its top-down control from other cortical circuits, such as, say, area 46. And with that, then, I'd just like to say thank you, all of you, for being here and for listening to my talk. But most importantly, I would like to thank, in terms of the lab, Professor Angela Roberts for kind of allowing me to pioneer a lot of this chemogenetic work, and importantly, everyone else in the lab, and Dr. Laith Alexander for really being the person to kind of drive forward the pharmacological overactivation work initially. Everyone else in the kind of Roberts and Clark labs working with the marmosets, our funders in the MRC and the Wellcome Trust, and then the BAP for funding this symposium, as well as also, most importantly, the marmosets themselves. Thank you.
Thank you, Christian. Do we have any questions for Christian? Yep, just at the front.
Thanks so much for such a great talk. I had a question, it might have been a misunderstanding, about the appetitive and aversive assays that you used. So is it after they've already acquired the conditioned response that then increasing activity in the SGACC then results in effect, or does it also disrupt the acquisition of a Pavlovian reward response, which I thought would be...
Great question, and that's perhaps myself for not explaining that particularly well. So that we train them such that they have a very stable discrimination of CS plus, they show the anticipation, CS minus, they don't really show anything. It's very much not associated with the acquisition of that process. It's very much once it's ingrained. That really they know that sound happens, they get excited, and we see it over the course of about 5 sessions during training. That's when we're happy to kind of cannulate and then do the kind of manipulations. So we do a lot in terms of control stuff to make sure that there's no other stress-related aspects impacting it. So yeah, it's definitely once it's well established.
That's really interesting. And have you ever tried it, you know, the other way around, where you're looking at the acquisition, any disruption in the acquisition?
We haven't. And I think the issue with that to a certain extent is this DHK acts for about 45 minutes, and it would be the case of what might the activation of 25 do within that period. Okay, it may diminish their kind of association or alter the kind of context in the environment. But yeah, we haven't necessarily done that. But it would be interesting to do, but my concern would be is that some of the other aspects related to threat might be in there that might actually prolong the acquisition. But I suppose that's probably the point really to kind of tease into that.
Yeah, no, really interesting. Thank you.
Do we have any more questions? Just at the front. Oh, at the back.
Thank you. It was really wonderful. My question is, have you looked into human subjects for the fMRI studies or something like that to correlate these findings?
So the work that we do primarily is through causally manipulating area 25. So we haven't obviously we can't really do such a manipulation in humans. Obviously there is aspects that there are neuromodulatory therapies that are now being utilized for more causal studies in humans that hopefully, I appreciate there was, I think, a symposium yesterday kind of along that topic, but perhaps there may be a methodology to stimulate area 25, thank you, in the future. So yeah, so perhaps we can get round to that and start to look at this more causal finding in humans in the future, for sure.
I meant the people who have PTSD and things like that, they would have some sort of threat and those things already activated. So possibly, I was thinking...
yeah, I mean, when I'm speaking to the idea that 25 is active, I don't wish to be specific about it's only depressed patients that might exhibit that phenomenon. As my kind of slide earlier on with all the lots of red all over the brain, there is certainly heterogeneity, and there are certainly probably aspects relating to this hyper-responsivity to a threatening situation that PTSD patients might show that might be correlated with the fact that they have an increased activity in area 25. It's been a while since I've looked at that literature, so forgive my ignorance there, but yeah, I'm pretty sure there's probably something along those lines, for sure. Thank you.
Thank you.
I think there was a question at the front, yeah.
Hi, sorry, I just had a really basic question. Just, do you see a difference between sex of marmosets, or are they all one sex that you're testing on?
Brilliant question. So we actually do males and females. So all the data is kind of agnostic to that to a certain extent. We haven't broken it down, but for an N of 4, it'll be 2 and 2. But we see it in both males and females. There's never really a sex difference, but these are quite big effects. So I'd be surprised if we could pull out perhaps a sex difference at that point. But certainly when, as we kind of grow and replicate these findings, it may be useful to kind of try and tease out whether there is a stronger one affecting, say, a female or a male. Yeah.
Very impressive. I wonder, because your findings reveal striking the association between area 25 projections to nucleus accumbens and the amygdala, each driving distinct aspects of depressive behavior. Do you think this circuit-specific model could inform symptom-specific interventions in depression or schizophrenia, for example, targeting anhedonia versus threat sensitivity separately? Thank you. A little bit nervous.
So speaking to the idea that, so we have this difference of this blunting of reward or anticipatory anhedonia akin to in the marmosets when we activate 25, they are actually differentially reactive to SSRIs don't actually impact this anhedonia effect in the marmosets, but that's acute. We haven't studied that chronically. But here I kind of have an indication of the fact that actually when ketamine given 24 hours before, it can actually blunt the area 25 mediated anhedonia. And that's up to about a week. So by about 3 weeks, this effect is kind of gone. But importantly, this heightening of threat when you activate area 25, ketamine doesn't impact it. And we've also looked at SSRIs, and I believe it's still ongoing to a certain extent, so I don't know what I'm supposed to say. But I don't think it is quite also able to ameliorate that heightening of threat response as well. Hopefully it is useful, but it's only one part of the giant jigsaw that is trying to tease out what symptom these drugs should hit. But I think there's certainly something that, there's another postdoc in the lab, Kevin Mulvihill, who's looking at different prefrontal manipulations and how they relate to this kind of heightened threat response. And he's definitely showing that depending on the neurobiological dysfunction that drives this heightened threat responsivity, that impacts what treatment might actually be responsive to as well. In the case of, I think it's between the aspects of the orbitofrontal cortex, between, say, SSRIs or ketamine, it kind of differentiates which drug you should utilize. But that's not necessarily really something that I think is going to be applicable globally in terms of getting these subsets of patients to, one, want to go and sit in a scanner for a while to get that data, but two, then think about changing their treatment strategy. I'm not sure. But thank you.